The central nervous system consists of the brain and the spinal cord. The first and foremost thing we need to think about these two key organs is their protection. Yes, they are protected by three different mechanisms, three different setups. First one happens to be the bony case. The cranium protects the brain, and the vertebral column protects the spinal cord. So what you see here is a part of the cranium. Okay. Then the second means by which the brain and the spinal cord are protected is by the meninges. Meninges plural, meninges a singular. Meninges are a set of connective tissue layers. The outermost is a dura mater, the middle layer is arachnoid, and the innermost layer is the pia mater. Okay, so let's talk about where they are in here. The thick layer that you're looking at that's shown in gray happens to be dura mater. Okay, dura mater is a very thick and a durable connective tissue sheet. This is the outermost layer of the meninges. Now what you see here is a number of capillaries, and those are referred to as sinuses. Okay, different names based on their locations, but we would not be learning those details. So dura mater, and then there is this orange layer underneath. That's what you call as arachnoid mater, or simply called as arachnoid. Then the innermost layer is shown in yellow here. This is pia mater. Pia mater is a very fine connective tissue layer right next to the brain or the spinal cord tissue. Although it may appear as a very thin layer, it is durable, it's tough. All right, so there's no space between the brain tissue or the spinal cord tissue and the pia mater. Then the space that you see between the pia mater and arachnoid is referred to as subarachnoid space, because it's underneath the arachnoid. You call it a subarachnoid space. It's a wider space. Okay, subarachnoid space is where you would see the circulation of cerebrospinal fluid. Well, between the dura mater and the arachnoid, there is this thin space that is shown in white here. That space is what you call as subdural space. Makes sense because it's right below the dura mater. So if you were to go in the order of a dura mater, and then the subdural space, then arachnoid, then you have subarachnoid space, and then the pia mater. We have one more space to add here, that is between the bony case and the dura mater. So it's outside of the dura mater. This space here, light gray color, is what you call as epidural space. Epidural space. Okay, so all these are discussed now. Epidural space, that is typically padded with adipose tissue and blood vessels and some connective tissue. Now then the dura, then the subdural space, arachnoid, subarachnoid space, and the pia mater. Okay, with these details, let's switch our focus to talk about the cerebrospinal fluid, where it's formed, what it does, and how it circulates or removed from the body. Okay, so in this view, we can still see quite a few things. You're looking at the spaces we have already looked at pia mater would be right next to the tissue. This is our subarachnoid space. Subarachnoid space is below the arachnoid, and then you have arachnoid, and then the dura mater is this bluish layer. Okay, and that's where you saw the blood vessels before, the capillaries, and so let's think about them as leading to veins eventually. All right, so if you recognize the meninges, now let's talk about the cavities that we have in the brain and in the spinal cord. The spaces or cavities that you find in the brain are called the ventricles. The ventricles are shown here. They are organized into different parts based on their locations. Okay, lateral ventricles, third ventricle, and then the cerebral aqueduct, fourth ventricle, which continues then as the central canal in the spinal cord. All right, this is our subarachnoid space. All right, so the spaces that you find in the cerebral hemispheres is what you call as the lateral ventricles. The space you find in the diencephalon area, that is in the region of thalamus, hypothalamus, epithalamus, that's what you call as the third ventricle. Then that continues as the cerebral aqueduct, and then becomes fourth ventricle. Cerebral aqueduct actually sits in the midbrain. Okay, this area is the midbrain, and the cavity that you see is narrow, and that's what you call as mesencephalic aqueduct or cerebral aqueduct. And now you're looking at the pons and cerebellum. The cavity here widens, and that's when you refer to that as fourth ventricle. Okay, so you have two lateral ventricles, one in the right cerebral hemisphere, the other one in the left hemisphere. Only one third ventricle, and they're all, from that point on, just one cavity. Okay, now let's talk about where cerebrospinal fluid is made. All right, what you're looking at here is a special arrangement of capillaries. These capillaries are special because they don't have this blood-brain barrier around them. All right, blood-brain barrier, as you may recall from the discussion in the previous places, is an additional layer around capillaries. Okay, that prevents leakage of unknown substances out of blood, and these unknown substances may cause harm to the brain or the spinal cord, and that's what is prevented by this additional layer, which is added by one type of neuroglia called astrocytes. Although all capillaries more or less have the blood-brain barrier, some special structures such as here are not, and that kind of arrangement of capillaries here is what you call as choroid plexus. So there is choroid plexus here, choroid plexus here, choroid plexus here. So if you're thinking about this as lateral ventricle, yes, there is choroid plexus. This is third ventricle, and there is choroid plexus. And then there is fourth ventricle, there is choroid plexus. This is where you have the capillary network, okay, bundles of capillaries that are without the blood-brain barrier. So now what happens is we can extract plasma out of these capillaries into these spaces. So the arrow showing the arrival of plasma from the capillary into this space. So cerebrospinal fluid, CSF, is simply a collection of plasma, okay? trapping of plasma, extraction of plasma into the ventricle. So it can happen in 3 different locations. One is lateral ventricle, then the third ventricle, and then the fourth ventricle, with the help of choroid plexus. That is the setup that exists. That is the arrangement that allows the brain to secrete or make cerebrospinal fluid. If you recall, there is another type of neuroglia called ependymal cells. Ependymal cells play a key part in the extraction of plasma. These are the cells, ependymal cells that line the ventricles. They're the ones that actively move the plasma from the capillary into the ventricles. All right, so now we have started making cerebrospinal fluid in the lateral ventricles, and it moves through a narrow opening. Okay, so you're looking at interventricular foramen, or foramen of Monroe. So that is how it gets into. It's a tiny opening that gets into the third ventricle. All right, from the third ventricle, where there is additional CSF added, the whole thing would continue through the cerebral aqueduct. From the cerebral aqueduct, it goes through the fourth ventricle, where additional cerebrospinal fluid is added. Okay, so here we have 3 openings, 3 apertures. Lateral aperture is one that is shown here. Basically, what happens is here the flow of CSF can take 2 paths. All right, the flow of CSF can be into the central canal, which is the continuation of the fourth ventricle, or depending upon the space that's created by these apertures, you have 2 lateral apertures and then 1 median aperture. So CSF can get into the subarachnoid space. Okay, so it can take 2 paths, one to subarachnoid space and another one to the central canal. So there is the distribution of it. Do you see how it goes into the subarachnoid space that takes cerebrospinal fluid around the brain or around the spinal cord? Okay, so now you can imagine the brain and the spinal cord surrounded by, both are surrounded by cerebrospinal fluid, which is a really good setup because the fluid around the tissue can absorb shock and protect the brain and the spinal cord. In addition to this role, cerebrospinal fluid can serve as a distributing agent. It can take nutrients and wastes and neurotransmitters from one place where they're made to other parts of the brain or the spinal cord. Okay, so gases and nutrients and wastes all can use cerebrospinal fluid as a medium to reach from one part of the central nervous system to another part. Absorption of shock and distribution are 2 key functions. All right, now let's talk about the amount of CSF that's made here and how that is replaced so frequently. Okay, approximately about 600 to 800 milliliters of CSF made per day. Okay, so that is all shown here. All right, production about 800 milliliters, and you're talking about at any one time you would see anywhere from 100 to 150 ml in circulation. Okay, it's constantly being made as it is constantly being removed. Okay, So per day, it's about 600 to 800 milliliters. What does it consist of? Pretty much everything that's in the plasma, because it's simply the movement of plasma into the ventricles, isn't it? So now you have sodium ion in larger amounts and chloride ion, and a very small amount of protein, and very small amount of glucose, very small amount of potassium ion and calcium ion, just like in the other places that you have studied, and um, mainly water. Okay, pH is almost neutral. All right, so let's go back here to talk about the production and the pathway through which it circulates. Basically, you have seen it already. It's just spelled out here. Choroid plexus in lateral ventricles synthesize some CSF. Foramen of Monro, this is the opening that leads to the third ventricle, where there is additional CSF added. And now cerebral aqueduct, also called as mesencephalic aqueduct, that is situated in the midbrain. It's also called as aqueduct of Sylvius. Through the mesencephalic aqueduct, it gets into fourth ventricle, where you make additional amount of CSF, and then foramen of Luschka and Magendie, these are the apertures we talked about as lateral aperture and median aperture, and it can get into subarachnoid space. We are ignoring the part that it continues as a central canal in the spinal cord, because we are looking at the absorption of cerebrospinal fluid. So absorption happens in the subarachnoid space. How? The amount of CSF that gets into the subarachnoid space will ultimately be drained into the capillaries. Okay, the arachnoid would have these finger-like projections here and there, and they're called arachnoid villi and arachnoid granulations, and they lead the emptying of CSF into those dural sinuses that we talked about before. So here is our arachnoid granulation, arachnoid villi. Those are the finger-like projections that drain CSF into these dural sinuses. All right, so that is how you remove the previously made CSF while fresh CSF being extracted from the plasma. Okay, so the fluid that you collect from the plasma is returned back to the vascular system. It's shown beautifully here. This is arachnoid space, subarachnoid space, the projections that you see. All right, you call it as arachnoid granulation villi or arachnoid granule. Okay, so that is how you drain it into the capillary, and you, as I mentioned, CSF is nothing but the extraction of plasma, and you return it back to plasma eventually. Okay, good. So that is all you really need to know about CSF, its functions, its formation, its circulation, and its reabsorption. All right. So the volume we already discussed, the amount that circulates, or the total amount that one can see within a day. Now, anytime there is a blockade of the circulation of CSF through the ventricles, which typically happens in the narrow spaces such as the cerebral aqueduct. Okay, when there is blockage, what do you expect? The fluid buildup within the brain tissue, it's not drained as usual, so the buildup of fluid increases the intracranial pressure. Increase in intracranial pressure would compress the brain tissue and also affects the functions of those structures that are affected. So that is a pretty dangerous, and then interventions are needed to correct the situation. All right, so this is where we stop. Thank you very much.